Hi, and welcome to my presentation on Chapter 23, Evolutionary Processes. So we've kind of talked about natural selection already, and we kind of mentioned gene drift and sexual selection, but in this chapter we're just going to go through all the different processes that cause evolution to happen or drive evolution in a bit more detail. So natural selection is of course the main one, or the most widely thought of one. In natural selection, you have changes in allele frequencies that promote survival of the individual, and so you'll have any alleles that give traits that are beneficial for survival kind of increasing over time in their frequency in the population. So alleles that increase survival will become more frequent in the population over time. Alleles that decrease survival will become less frequent in the population over time. And I should add that it's not really just survival that we're looking at there, it's fitness, which is contributed to by survival. So fitness is the amount of offspring you have, and they have to be viable, fertile offspring. So offspring that live and offspring that will be able to have offspring of their own. So if you're surviving for longer, you're probably going to have more offspring. If you don't survive for very long, you're not going to have many offspring if you have offspring at all. So in addition to natural selection, we also have genetic drift driving evolution, which is just random changes in the allele frequencies. We have gene flow, which is changes in allele frequencies that are caused by individuals moving from one area to another, or between one population and another population of the same species. You also have mutation, which is how you create new alleles in the first place. You have sexual selection, in which you have changes in allele frequencies that are caused by mate choice or by competition for mates. So sexual selection can actually be considered sometimes a type of natural selection. Like some books will consider it a type of natural selection. I'll consider it separate from natural selection because a lot of the time traits that are helpful for finding a mate or being attractive to a mate or competing for a mate are not actually helpful for survival, and they'll actually hurt your survival. So it's possible for sexual selection and natural selection to kind of be in opposition to each other, or for them to be kind of promoting different traits in the population. So we'll consider them separately. And then finally you have artificial selection, which is changes in the allele frequencies caused by mating that's directed by an outside source, so selective breeding. Usually that would mean like human beings are selectively breeding individuals in a population to get certain traits that we want in the offspring. And then finally, lateral gene transfer, also called horizontal gene transfer, can also cause evolution or contribute to evolution, that would be where you have genes transferred from one organism to another, but not by heredity, so not by like descent, not one organism giving birth to another organism, but unrelated organisms transferring genes between each other. That would be common in microbes or microorganisms. So we're going to go through each one of these processes in turn in more detail. We'll start with looking at the Hardy-Weinberg principle, which was developed by a couple of scientists, G.H. Hardy and Wilhelm Weinberg. They were trying to predict what effect random mating would have on the allele frequencies in a population. So they created this concept called the gene pool, which is actually an imaginary thing. It doesn't exist in real life, but it's useful to think about. So the gene pool, which one is shown up here, it contains all of the alleles for a given gene in a population. So every individual in this population would have 2 copies of every gene. So for this particular gene, let's call it gene A, there's 2 different alleles that are possible, big A, little a. Every individual would have either 2 big A's, 2 little a's, or 1 of each. But in the gene pool, we kind of ignore that. We ignore the individuals, and we just look at the alleles that they have separate from each other. So we take all the alleles If you were AA, big, or I should say, if you were big A, you would contribute 2 big A's to this. If you were big A, little a, you would contribute 1 big A and 1 little a to this. And we just kind of mix all these alleles up as the total number of alleles for this gene in this population. So that's the gene pool. Then we're going to look at how these alleles could potentially combine with each other if you had random mating. So for the big A allele, we'll denote that using P. We'll say that P is the frequency of allele big A. So in order to figure that out, you would count up all the total alleles that you have here, both the big A's and the little a's, and figure out basically what proportion is big A's. And you have in total 40 alleles there, 28 of them are the big A. So when you divide 28 by 40, you get 0.7, and that's P. So the frequency of the big A allele, or P, is 0.7 in this particular population, or in this particular gene pool. And then you can do the same thing with the little a allele, but we'll use Q to represent little a instead of P. So the frequency for little a is going to be 0.3, or Q would be 0.3. Then we'll basically make a Punnett square to see all the possible combinations between these alleles if you had random mating. So you could have big A combining with big A, little a combining with little a, or big A combining with little a. And there's 2 different ways to do that. It could be big A from the mother, little a from the father, or it could be big A from the father and little a from the mother. So this is a basic Punnett square. The difference is that we're going to also take into account the frequencies of these alleles, so the P and the Q values. So to get the actual frequency of big A, of the offspring that are big A, or homozygous for the dominant allele, we would times P by P, so we get P squared. To get the frequency of the recessive homozygote, the offspring that have little a, we would times Q by Q, and that gives us Q squared. To get the frequency of the heterozygotes, we would times P by Q, and then you have to do that twice. So it would be P times Q plus P times Q, or 2PQ. So those are the frequencies of the genotypes of your offspring. And you can add all those numbers up. So you would add up P squared, add up 2PQ, and add up Q squared. And all of these numbers are going to equal 1 if you just had random mating there. So that means basically you could use that equation, P squared plus 2PQ plus Q squared equals 1, to figure out the frequency of P and of Q in the offspring generation. And you would find that P still equals 0.7 and Q still equals 0.3. So basically there's no change there. So the upshot there is that if you have total random mating in a population, there's going to be no change in the allele frequencies for any of the genes that you look at there. Which means evolution is not happening, because for evolution to happen, you have to have allele frequencies changing between the parents and the offspring, but that's not happening here. So the usefulness of the Hardy-Weinberg equation there, the P squared plus 2PQ plus Q squared equals 1, is that we can use it to kind of figure out if evolution is happening for a given gene in a given population at a given point in time. So you would measure the allele frequencies for all the alleles for that gene in the parents and in the offspring, and you would use that equation to see, like, do the allele frequencies in the offspring actually match the predicted results of this equation? Do they actually all add up to 1? If they don't add up to 1, then evolution is happening. If they do add up to 1, evolution is not happening. So when Hardy-Weinberg did this kind of experiment of theirs, they were assuming in their random mating setup, they were assuming no mutation is happening, no natural selection is happening, no gene flow is happening, no gene drift is happening, and there's no sexual selection or artificial selection, because that would mean that there's no random mating. So if you kind of look at a real life population and you see that their equation holds true, you know that all those assumptions must be met. There's no mutation occurring, no natural selection, no gene flow, no gene drift, no sexual selection, and no artificial selection, which means evolution is probably not going on for that gene at that point in time. If you get that equation holding true and the allele frequencies in the offspring generation are equal to 1, once you add them up by that equation. If that's not the case, if you were to plug in the allele frequencies in the offspring for P and for Q and plug in that into the equation and figure it out, and you don't get 1 as your answer, let's say you get 1.5 or 0.5 or something like that, then you know that at least one of those Hardy-Weinberg assumptions is not accurate for this population. You don't know which one, but something is going on there that would cause evolution. So you don't know if it's natural selection or if it's gene flow or if there's sexual selection or mutation, but you know at least one of those things must be occurring in order for you to get a result other than 1 for that population. And the last thing we'll talk about before we get into specifically all those processes that drive evolution is inbreeding depression, because it's something that's going to come up when we look at some of these processes. So inbreeding depression is basically the reduction in fitness of a population if you have a lot of inbreeding in the population. And there's a couple of reasons why this can occur. If you have inbreeding, that means individuals who are related to each other are having offspring together. It's going to increase the chances of getting homozygotes. Right, if you're having offspring with a close relative of yours, you're genetically similar to them. It's likely that they're going to have the same alleles that you have for a lot of the genes that you guys share. So your offspring would probably have those same alleles. Instead of being a heterozygote and getting one allele from you and a different allele from your partner, they would get 2 of the same allele because you and your partner are related to each other. So when you have more homozygotes, for certain genes, it's actually not a good thing to be a homozygote, especially when it comes to genes in the immune system. It's actually good to be a heterozygote. Not sure entirely why that is, but for some of those genes in the immune system, they are related to the amount of different antibodies that your immune system can produce. So if you have a lot of different alleles there, you're going to have a lot more antibodies that you can make against different pathogens. If you have less alleles because you have more homozygotic genes, you're going to not be able to make potentially quite as many different types of antibodies. So you would have a weaker immune system, basically. It's also a problem because it can lead to the frequency of recessive genetic conditions increasing. So for a recessive genetic condition, that's a condition where in order to actually suffer from the condition, you have to have 2 copies of the allele that causes that condition, because that allele is recessive. So if you get 1 recessive copy of the allele, 1 dominant copy, the dominant copy is the good one, it's going to express, you're going to be fine. You're not going to have a disease, even though you do carry the allele for that disease. Because it's recessive, it's hidden. If you had 2 copies of that recessive allele, though, in that case you would have the disease. If you have a copy and a relative of yours also has a copy because you 2 are related, and then you 2 have children together, it's more likely that your children would get 2 copies and therefore would suffer from the disease. So because of things like this, a lot of inbreeding in a population is going to lead to less fitness in that population. There can sometimes be an upside a little bit, which is just that for those recessive alleles, if they're really deadly, if the condition they cause is really deadly, but only people will have just 1 copy, so they don't actually have the disease, they can just pass that on. It doesn't affect them at all. But if people are getting 2 copies a lot, those people are probably going to not have kids. They're going to not pass that on, most likely, if it's really a serious condition that's very deadly. So in some cases, it could actually help to eliminate some of those genetic conditions if you did have inbreeding. But usually it's a bad thing. Well, I mean, even in that case, it's still a bad thing for the people it actually happened to, for the organisms it actually happened to, right? um, so inbreeding is more likely to occur if you have a small population. There's just fewer options available for mates, so it's more likely that your mate will be related to you. It's also more likely if one individual is having a large number of mates. And we've got a couple of examples here. On the bottom here we have a graph showing survivor of panther offspring. So in Florida, they have some panthers or mountain lions. Not very many, it's a small population. They're definitely endangered because of habitat loss. Because there's so few individuals left, they have ended up inbreeding with each other a lot. And their offspring do not survive very well. So in red here, you see basically how many offspring are surviving from panthers who mated with each other, like Florida panthers mating with other Florida panthers. And there's a lot of inbreeding when you get that kind of cross, that kind of mating pattern. So their offspring are not really surviving for very long. Only around, what is that, like 25% are making it to their first year. And it just gets worse after that. By like 10 years, virtually all the offspring are dead. So one thing we've done to try and kind of support those populations and bring back the Florida panthers is bring in some more panthers from Texas that are unrelated, to bring in, new alleles to the population and reduce the inbreeding that's there. So when you have a Texas panther meeting with a Florida panther, you can see the survival of the offspring is way, way stronger. Like half of the offspring are surviving to their first year, and by the time you get to 10 years, still like around 45% or so of the offspring are alive. So the survival is way, way better when you when you reduce the inbreeding there. On the top here, we have a family pedigree for a family in was it Short Creek, Arizona and Utah. And this is a family where you have a lot of individuals suffering from fumarase deficiency. So in this pedigree here, men are represented by squares, or fathers are represented by squares. Women or mothers are represented by circles, and you can see if a line goes between a square and a circle, those people mated and they had their offspring. In this case, this particular couple had one daughter, and then she went on to have offspring with this man. They had one son. By the time you get down to the 6th generation here in this family tree, you're starting to see some of these squares and circles be filled in. Those are individuals who are suffering from fumarase deficiency. Fumarase is an enzyme in the Krebs cycle, so it's really crucial in order to be able to make ATP that you have that enzyme and it functions correctly. If you have fumarase deficiency, that is a recessive condition. So if you have just one allele for the fumarase enzyme that's kind of broken, that's fine. You're going to have another copy that works. That one will be dominant. You'll be fine. If you get 2 copies of the broken fumarase allele, then you're going to have this fumarase deficiency condition, and it's a very serious condition, as you might imagine. People who have this condition will not ever make as much ATP as someone who doesn't have it. A lot of the time they die like very young. If they don't die, with enough care they can survive, but they're probably not going to talk or walk or feed themselves ever in their life. This is an extremely rare condition. When they found this kind of cluster of individuals that all have it in this one community, like at that point in time, only 13 cases were known anywhere in the world. And when they found this cluster, it was like 20 different people who had the disease. So right in that moment, the number of people who were known to have the disease worldwide, like over doubled, more than doubled. And the reason why you had so many people with this disease in this community is because this is a small community where you have polygamy going on. So you'll have one man taking multiple wives. And that was kind of how the community was founded. The community follows the FLDS Church, which is the Fundamental Church of Latter-day Saints. So they're basically Mormons. The original Mormon teaching includes polygamy, but that kind of conflicted with the U.S. government, which had outlawed polygamy. So the Mormon Church officially moved on from that practice, but you had some members of the church who weren't ready to give it up because it was their religious teaching. So they kind of splintered off from the main Mormon Church and founded the FLDS. And a couple couple members of the FLDS founded this community, Short Creek, in Arizona and Utah. It's kind of on the state line. And you had some of those founding members who are basically related to almost every person in that community today. Like a couple of founding men in that, for that town, like 85% of people who live in that town are genetically related to those two men. So you can see by the time we've gotten like 6 generations on from our starting couple here, there's a lot of inbreeding going on. There's a lot of cousins marrying other cousins and having offspring. So probably what happened here is this great-great-grandfather here had a recessive copy. He had a recessive fumarase allele, the defective copy. He did not have a disease because he only had, one bad copy. He had another good copy, but he passed that recessive defective copy on to his offspring. And a few generations later, his offspring are all marrying each other, breeding with each other, and their offspring are getting 2 copies. One from their mother, who got it from her great-great-grandfather, and one from their father, who got it from her great-great-grandfather, or who got it from his great-great-grandfather. And so you have a lot of individuals with this disease in this community now. That actually was in some way beneficial for the whole human species, because we had kind of understood that this disease, fumarase deficiency, was genetic, but we didn't really know like what gene causes it, what's really going on there. And it was really hard to study because it was so incredibly rare. When we discovered this cluster of individuals with the disease in this one community, and we know how they're all related to each other, and we can look at their genetics, then we were able to actually identify what gene causes the condition. So it's possible that in the future, we might even be able to cure it with that information if our genetic technology, kind of continues to strengthen over time. So that brings us to natural selection. So there's a few different ways that natural selection can occur, or I should say a few different kind of patterns that can be observed, or different types of natural selection, which we'll go through here. So first you have directional selection. In directional selection, the average value of a trait is changing to a new value, so it's either moving up or moving down. Maybe you're getting bigger, maybe you're getting smaller, something like that. So that means the current value is not very adaptive. The new value that you're moving to is more adaptive. This type of selection is going to overall reduce genetic diversity. It's going to make all the individuals more similar to that the new value for the trait. You also have stabilizing selection, which means that the current average value is actually the most adaptive value for the trait, and everybody is slowly over time just becoming more and more close to that ideal average value. So you're reducing the overall variability in that particular trait, and therefore also reducing the genetic diversity there. You can also have disruptive selection, where the average value for the trait is actually the least adaptive value. So it would be good to have a bigger value or a smaller value, but not good to have the average value. This is going to increase the variability in the trait, especially at the 2 extremes, the biggest values and the smallest values. It's going to at least maintain genetic diversity, but can actually increase genetic diversity and can even lead to speciation over time. Finally, you have balancing selection, where you're maintaining the current variability in the trait, which means that there's multiple different values for that trait that are all equally adaptive to each other. So not just the average value is the most adaptive, but values that are above or below the average are also equally adaptive as the average value. So this is also going to either maintain or even increase genetic diversity. A lot of the time you'll see balancing selection happening when heterozygotes are favored. Like as I mentioned with immune genes especially, heterozygotes will typically have a stronger immune system than a homozygote for crucial genes in the immune system. We call that heterozygote advantage. So that's a form of balancing selection. You also have something called frequency-dependent selection. That can also be a type of balancing selection. So in frequency-dependent selection, an allele is going to be more adaptive if it's rare, but if it becomes common, then it becomes less adaptive. So the best example of that would be like coloration patterns. and how that interacts with predators. So predators are looking for their prey. They know what their prey looks like they know what the coat pattern for the prey is going to be. So if they see that coat pattern, they're going after that prey. Excuse me. So what happens if you have like a rare pattern to your coat? That's actually adaptive for you. The predator doesn't recognize that rare pattern, so it doesn't know to look for you. It doesn't really recognize you as a prey animal as quickly, so it's going to ignore you more commonly. So now you have this advantage. You're going to live longer. You're going to have more children, and that rare coat pattern that you had is going to become more common. Right, so over time, as you're having more and more children, well, maybe not you anymore, but the number of individuals in the population with that rare coat pattern is increasing. The predator is going to catch on. It's going to notice, well, a lot of my prey kind of look like this now, and it's going to start hunting you as well. So now that coat pattern that used to be an adaptive trait for you is no longer an adaptive trait because it's become common. So that's frequency-dependent selection, and that's a type of balancing selection. Okay, so here we have directional selection again. This is where the traits are changed. Well, the average value for the trait is changing. It's moving either up or down from the previous average value. So the starting average value is less adaptive. In this case, as it's shown in this graph, it's better to be above the average value. So over time, the whole population is going to shift to this new average value that's above the previous one. And the variability in the trait is going down. It's kind of this hump here is wider in the first graph. This is representing the variability in the trait between how many individuals have a low value, how many individuals have a high value. And you can see after selection, after directional selection, that variability has gone down. An example of this happening is with finches in their beak size after a drought. So you have, this would be the distribution of beak sizes. Excuse me. So here you have in kind of paler orange, you have the original distribution of kind of beak sizes in this population of finches. So the average value would have been right here. Then you had this major drought that went on for a couple of years. A lot of finches died. The ones that were left, on average, had a bigger beak. So the beak went from smaller to being bigger. And that's probably because the seeds of plants that actually survived through the drought were kind of tougher, bigger, harder to crack, and the big beak finches were better able to get at them. So they suddenly had the more adaptive value for beak size. And the overall beak size of the population increased during that time through directional selection. You also have stabilizing selection, where the amount of variability in the trait is going down and the current average value is the most adaptive value. So you would go, the average doesn't change, but you would go from more variability in the trait to less variability in the trait. A good example of this is actually human birth weight. If you have a very small baby, it's less likely for that baby to survive, just because they're so small. Their body has so few resources. On the other hand, if you have a very large baby, it's also unlikely for that baby to survive. What's going on is that it's actually very dangerous to give birth to that type of baby. So it's likely that both the mother and the child will die during childbirth if the baby is extremely large. So what's best is to be in the middle. And so human birth weights have been kind of maintained at around 7 pounds for, centuries, really. Well, probably longer, right? Thousands of years, millennia, due to stabilizing selection. You also have disruptive selection. So this is the first type of selection that can actually increase the amount of variability that you have in a trait, or will at least maintain it. So increasing the genetic diversity or maintaining it. So in disruptive selection, you're starting with a population where here's your average value for the trait, here's your variability, and all of a sudden this average value is not a good thing to have. It becomes maladaptive. So it's good to be well below the average or well above the average, but it's not good to be average. So you're going to get kind of two humps forming. All the individuals who have a high, a high value for the trait, all the individuals who have a low value of the trait, and less individuals with the former average value for that trait. This can lead to speciation if these two groups kind of just totally split off from each other and stop breeding with each other. So an example of this in action is the gill rakers in whitefish. So whitefish in their gills will have these little projections which are called the gill rakers. They're actually used for feeding, for filter feeding. So as you're swimming through the water, your food will get stuck in those little gill rakers and you can eat it. It's good for whitefish to have, either a high number of gill rakers or a low number. It's not good for them to have a medium number. If you have a high number like this, they're going to end up really closely spaced together, and you're going to be really good at filtering out super tiny food from the water, like little planktons and stuff. If your gill rakers, if you have fewer of them, they're going to be more widely spaced. Tiny food is just going to pass like right through there. You're not going to filter it out at all. But the benefit of having like fairly widely spaced gill rakers here is that you can actually filter out like kind of little animals, like larger prey from the water. If you have a medium number of gill rakers, then you don't have enough to filter out the little guys. And when you go to filter out the bigger animals that are in there, your gills will actually just get clogged really quick. It's better to have, you want a filter that's sized appropriately for the thing you're actually trying to filter out, which would be this, this size here. So that kind of has resulted in two populations of whitefish, the ones that have the few gill rakers that are widely spaced, and the ones that have many gill rakers that are closely spaced. They're using totally different food sources, and there's a good chance that they're going to speciate if they go on long enough, because there's no benefit to mating. If you're one of these guys with a low number of gill rakers, you wouldn't want to mate with one of these guys that has a high number, because what if your offspring come out with a medium number? Then that's not going to be good. You want to mate with someone else who has a low number like you, so your offspring can be good at eating the type of food that you eat. Okay, then we'll look at sexual selection next. So in sexual selection, it is selection based on your ability to attract a mate or compete for a mate. So based on courtship of some type. So a favorable trait here would be any trait that increases your success in courtship, which might be a trait that actually harms your overall survival. So sexual selection can be opposed to natural selection. There's two basic types of sexual selection. You have intersexual selection. Inter means between, so intersexual is selection between the sexes. So that would be basically attracting a mate. You also have intrasexual selection. Intra means within, so within the sexes. So that would be basically competing for mates with other members of your own sex. Both types of sexual selection, intersexual and intrasexual, will result in sexual dimorphism in a species over time, which is where you have a difference in the traits between males and females for a given species. Like for instance, it's really common for males to be bigger than females. That's a form of sexual dimorphism that's caused by intrasexual selection, where males are competing with each other for mates, and the bigger male will usually win. So an example of sexual selection and how it can kind of be opposed to natural selection would be peacocks. So this is a female peacock, this is a male peacock. You notice the male peacock is a lot more colorful, even when his tail is kind of down, but when he puts his tail out, it's this gigantic, beautiful thing. So this tail is for intersexual selection. The purpose of the tail is to attract the female. She likes the look of the tail, and if you have a nice, big, impressive tail, maybe she'll pick you for a mate. So having a big, impressive tail is going to increase your chances of success during courtship. But when it comes time to living your life and surviving outside of courtship, this is not a good thing to have. It's gigantic. It takes a lot of resources to build these giant feathers and put all the pretty colors in them. And then when you have it collapsed, it's like trailing behind you on the ground. So this is going to slow you down. If you're running away from a predator, you're going to be slower. It's easy for it to get caught and stuff, and the predator can just come and step on it. So if you are a male with a giant, impressive tail, you spend a lot of resources on that, and you're more vulnerable to being caught by predators. So it's actually not good for your survival to have that. But over time, as it turns out, you will actually have more offspring because the females like it. So sexual selection usually does not affect both sexes in the same way. It usually will act a lot more strongly on males than on females. This is because of the difference in resource commitment to reproduction that males and females have. For females, they produce eggs. An egg is always much, much bigger than a sperm and a lot more expensive to make. So here you can see an egg, and in blue on its surface you have a bunch of different little sperm. So that's the body of a sperm and then the tail coming off of it. So you can see like a sperm is frickin' tiny compared to an egg. There's so little resources in every sperm compared to the amount of resources in that egg. You're going to invest way more resources to make eggs than to make sperm. Then on top of that, usually in the animal kingdom, it's the female that will actually be pregnant and, will carry the young after fertilization. Usually it's also the female that will provide the majority of childcare, any childcare that is provided by that given species, which some species, of course, don't really provide childcare. That's on average. Sometimes it's not like that. Sometimes the male is providing more parental care. Occasionally it's even the male who's the one who gets pregnant, like in seahorses. It's the male who carries the young. But usually it's the female who's investing those resources to actually build the body of the new offspring and take care of it once it's born. So for females, their reproductive success, how many offspring they can have over their lifetime, is mostly limited by their access to resources. How many resources can they get access to build those eggs, to be pregnant with offspring, to take care of the young? That requires a lot of resources. For males, they don't invest nearly as many resources in reproduction, usually. And so usually for a male, for most species, their reproductive success is limited more so by how many times they get to mate, how many females they're able to mate with. So that means that for a female, in order to increase her reproductive success, she's going to be focused on obtaining resources, picking a mate who's going to be genetically fit, who maybe will help her get resources. For a male, in order for them to be reproductively successful for most species, they're going to be focused on having a lot of mates, having as many mates as possible. So this means that probably it'll be the female who's being picky about who she mates with. The male will not be very picky, and probably males will end up competing with each other for mates. So this means that sexual selection will overall be working more strongly on males. They're going to be the ones who are trying to attract a mate and the ones who are competing with each other for a mate, usually. So if we look at intersexual selection and mate choice, what the female is looking at there is usually going to be either looking at the health of the male or the genetics of the male or how good they would be as a parent. So for instance, for birds, where you have a lot of sexual selection and a lot of intersexual selection where males are trying to attract females. The males will frequently have really pretty colors. So for instance, this is a male, I forgot which type of bird this is, but this is the female of the species and this is the male. Obviously a huge difference in what they look like and who's more flashy. All these beautiful colors in the male's feathers, those are coming from carotenoid pigments. Those carotenoid pigments are found in plants. So he's getting this from his diet. So when the female sees a male who's really pretty like that, she knows that he is getting a good diet. He's good at getting access to food. So maybe that means that he has good genetics. He's certainly in good health. Also, like, he looks really flashy to predators as well. So he's lived his life this long looking all flashy like that and not getting eaten by a predator. So maybe he's also really good at avoiding predators. Maybe that also speaks to his good health and his good genetics. So if she picks the flashiest male possible, there's a good chance that she's picking a male who's in good health and probably has quality genetics to contribute to her offspring. She wants to make sure she mates with, a healthy male, because she's going to invest so many resources in those offspring. There's really no point if they're just going to be sickly and die anyways. Another thing that males will do a lot for birds is they will actually contribute to parenting of the children or of the offspring. So they'll stick around. They'll help in some cases, like keep the eggs warm. Sorry. So this is a male penguin warming his mate's egg there. They can also help defend the nest. They can help bring food to the offspring once the offspring are born. So the males can have a major role to play in parenting as well. The female wants to make sure that she's picking a male who's going to contribute as many resources as possible and do a good job in that role. So she can do that in part by like looking at courtship dances. A lot of the time there'll be little courtship displays where the male will actually build a nest for the female, and so she can kind of judge, like, he made a really nice nest, he's probably going to be a good dad. So stuff like that. This graph here is an experiment where they took some females and males of the same species of birds. For the males, they actually had siblings, male siblings, so brothers, and they fed them different diets. So for each pair of brothers, one brother got a diet that didn't really have many of those carotenoid pigments. The other brother got a diet that did have a lot of carotenoid pigments in it. So you end up with a male who's really bright, who got the diet high in pigments, and another male, his brother, who is kind of dull. His feathers are dull, his beak is dull, because he didn't have a lot of pigments in his diet. And then we release these pairs of brothers into a cage with a female, and we see, where does the female go? Which brother does she prefer to spend her time with? The darker orange bar is the amount of time that was spent with the brighter brother, and the paler orange bar is the amount of time that the female spent with the dull colored brother. So you can see that they did this with 10 different females. For almost all these females, they're preferring to spend their time more so with the brother who has bright colors. Except for this one. No, lady number 9, she preferred the dull colored brother actually. So it doesn't work 100% of the time, but usually the female will go for the brighter male. You also have intrasexual selection, which is competition within the sex. So that would be competition for mates, usually males competing for females. Typically, this means that males will be larger than females because, the bigger male is more likely to win the competition. A lot of the time, these guys aren't really trying to hurt each other because, if you have like a serious fight with someone and there's not a hospital and you're both trying to survive, like, there could be serious outcomes for that. Like, it's not good for anybody to get injured. And so you'll just kind of do this thing where you like size each other up, and whoever's bigger, you'll just assume that that dude was going to win the fight. Or if you actually do fight, well, the dude who is bigger probably wins the fight. So a lot of the time males will be bigger because of that, and the biggest male will have the best chances for finding mates. In addition to that, sometimes for some species, males will have specialized structures that are for competition. That would be like antlers in deer. Female deer also do have antlers, but they're super tiny, and so you pretty much just forget that they're even there. They're just little nubs. But for male deer, they have big, beautiful antlers that they use to compete with each other for mates. So if you're successful in that competition, that might mean that you have really strong reproductive success and you find a lot of different mates. If you're unsuccessful in that competition, it's going to be hard for you to find a mate. You might find one, but you might find none also. So for those males who are very successful in these competitions, they can actually have a really large impact on the genetics of the next generation because they're having so many different mates, so many different offspring have like that one guy as their father. So this graph here is actually showing that for elephant seals. These guys are elephant seals. So these would be two male elephant seals competing with each other for mates, and they're actually fighting. It's pretty bloody. The bigger one will probably win there. So on the bottom you have reproductive success for female elephant seals. On the x-axis there you have the numbers of offspring that each female elephant seal has over the course of her life, and then on the y, how many females had that number of offspring. So you can see here like 60% of females don't even have a single offspring over the course of their life. So they have zero reproductive success actually. About 40% of females will have between 1 and 10 offspring over the course of their life. On the top here we have the same graph, but for males. So you can see like 90% of males are having no offspring at all throughout the course of their life. So the females, even though like a lot of females do not have reproductive success, overall the females are more reproductively successful than males. Because like the average male elephant seal isn't going to have kids at all, ever. For the elephant seals, the male elephant seals who are having kids, a few of them are having 1 to 10, a few more having 11 to 20 over the course of their life. There's a few that are actually having a lot, 41 to 50 kids, and a few, again, that are having a huge amount, 81 to 100 offspring. So these would be the males that are very successful at these types of competitions for mates. And they're having a massive impact on the next generation. So that's good for that individual male. He's very successful, having, 80 kids. But that's not necessarily always going to be a good thing for the population. If the population is very large, then this is probably fine. But if it's a small population, I mean, if we're talking about a population of only maybe 500 seals in the next generation, and 90 of them all have the same father, you're going to have a lot of inbreeding going on in that population pretty quickly from this type of mating pattern. So it can actually be a bad thing as well. Okay, next we'll talk about genetic drift, which is random changes in allele frequencies over time. So this would be caused due to randomness in who manages to find a mate. So sometimes, even though you're the biggest, flashiest male, you just don't happen to run into any females and you don't find a mate, even though, if you found one, she would probably pick you, and if you found another male, you'd probably successfully compete with him. Sometimes that happens. It's just random. Also randomness in deaths. So if you have like maybe a disease or a storm come through an area and kill just a bunch of individuals at random, those things are going to cause genetic drift. Over time, genetic drift is going to reduce the genetic diversity in a population. It's going to have a much stronger effect on a small population than a large population. But over time, even if the population is large, it will still reduce genetic variation eventually. It's possible actually for alleles to be totally lost by genetic drift. So all copies of that allele are gone, which would mean that all individuals who had that allele either died or just did not have offspring. Well, didn't have offspring before they died. It's also possible for an allele to become fixed, which means that it becomes the only allele for that gene that is left. All other alleles for that given gene are lost, and only that one is left over, so it's fixed. This is not a good thing. Especially if you're talking about a small population, those types of effects can occur quite quickly and really damage the genetic diversity of a population. And for endangered species that we're trying to conserve, genetic drift is a major barrier to that conservation because of the effect it has on genetic diversity. In addition to inbreeding, of course, being a problem. So these graphs on the side here are showing the influence of population size on genetic drift and how quickly it occurs. So on the top graph, this is basically computer modeling of the impact of genetic drift on a population. And we're looking at the frequency of allele A. So each one of these lines here is a different population, actually. All these populations have a size of 4. So there's 4 individuals. Every one of these lines here is representing 4 individuals in a population, and we're looking at the frequency of allele A. So if we look at the red line here, A drifted to fixation within less than 20 generations. On the x-axis, you have generations. So we started out with allele A at 0.5, so 50% of all individuals, all those 4 individuals had, allele A. And by the time you have had 20 generations pass by, in this particular population, A became fixed. It was the only allele that was left. In the blue line, we see A being lost instead of becoming fixed, so it's just gone. By 20 generations, it's entirely gone. Other alleles would still be present for that gene, but not allele A. So this is the same basic computer model. The only difference is the population size is 400 now instead of 4. So again, every one of these lines is one population with a size of 400 individuals in it. And we're looking at the frequency of allele A. It always starts at 50% or 0.5, and we just see what happens over time as the generations pass by due to random effects. And we see that in all of these populations, actually, not just the blue one and the red one, you still have allele A. It's not lost, and it also does not become fixed. In some of these populations, it's trending up towards fixation. Like in a couple of these guys here, it ends up after 100 generations being, 80, representing about 80% of all alleles for that gene. For the red population, it actually didn't change very much. It looks like it's still roughly 50%, maybe a little higher, maybe 55. For the blue population, it was trending down, so trending towards loss. But it still is, there's a good amount of A there. It's maybe, 30% or so. So in none of these populations was A actually lost or did it become fixed because of the population size, even though a lot of generations went by. When the population is very small, A was actually either lost or became fixed in every one of these populations before 20 generations had gone by. So we'll look at a couple of types of genetic drift. One is the founder effect. This happens after a founder event. So a founder event is when a few individuals from one population move to a new area and make a new population. So they're the founders for that new population. The founder effect is when that founder event means that you end up with different allele frequencies in the new population compared to what you had in the original population. So usually if you had a founder event, there's going to be a founder effect. So this is an example of that. Here you have your original population, a few individuals, in this case 2 homozygous dominants and 1 heterozygote, moved to a new area and founded a new population. So these individuals moving to this new area, that is the founder event. The founder effect is the fact that if you look at the genotype for these individuals, it's 2 homozygotes, dominant homozygotes, and 1 heterozygote, no recessive homozygotes. So the genotypes are actually different compared to the overall original population, which means the allele frequencies are different compared to the overall original population. In this new population, there's going to be less copies of the little a allele, the recessive allele, and more copies of the dominant allele. That's due to random chance. Just randomly which 3 individuals happen to found this new population. So that's the founder effect. Usually they'll go together. They don't necessarily have to, but they usually do. Technically, if you have equal amounts here of the heterozygote, the dominant homozygote, and the recessive homozygote, and you would have, if you had 1 individual of each group move to that new place, then you would have had a founder effect, a founder event, but you would not have had a founder effect. You will usually see this type of thing going on like islands and stuff, or other areas where the habitat is really isolated, and so it's hard to move there and hard to move back again. Another common type of genetic drift is the genetic bottleneck, which happens following a population bottleneck. A population bottleneck is where you have a large population and all of a sudden there's a catastrophe of some type and a bunch of individuals die and you're left with a much smaller population afterwards. The genetic bottleneck is where that population bottleneck causes the loss of a lot of alleles from the population. So that's another type of genetic drift. So again, here's your kind of example of that. Here's your original population. You have a bunch of dominant homozygotes here in this original population. Then we have some type of catastrophe happen. Maybe a major storm comes through or a disease. Almost everybody dies. So you have a population bottleneck where very few individuals are able to survive that catastrophe. And if you look at them, it just so happens that it was one of each type. One recessive homozygote, one heterozygote, and one homozygous dominant individual. Whereas in the original population, the most common genotype was homozygous dominant. So all of a sudden, just due to random chance, based on who randomly was able to survive this catastrophe, you have a genetic bottleneck where the frequency of the dominant allele has gone down and the frequency of the recessive allele has gone up. So this actually happened on an island called Pinjalap Atoll. I'm not sure exactly how to pronounce that. You had a typhoon come through that island and kill almost everybody actually who was living there. Only 20 people survived the typhoon. One of them happened to carry a recessive allele for a genetic disease called achromatopsia, which is a disease that affects your eyesight. Gives you color blindness and kind of just weak vision overall. And one of the effects is that you're really sensitive to bright lights. And so you can see a couple of these ladies here are squinting, probably because they suffer from achromatopsia and bright light, like what you would have just outdoors during the day. It's a lot for them, so they squint to reduce the amount of light coming into their eyes. So that disease used to be really rare on that island, and now it's really common because one of the 20 survivors happened to carry the gene for that disease. And next we'll talk about gene flow, which is the flow of alleles or genes between two populations caused by individuals moving from one population to the other and then breeding in that new population that they move to. So gene flow is going to cause those two populations to become more similar to each other genetically over time. That's not necessarily meaning that genetic diversity is decreasing, though. So you can see that in this kind of diagram here. Here's your first population, here's your second population, and we see that it so happens that in this first population, you only have homozygous dominant individuals for this particular gene. In the second population, you only have homozygous recessive individuals. So it's not very likely that that would actually occur in nature, but that is, you know, that's what we're showing in this diagram. You could certainly have a population where it's dominated by homozygous dominants or by homozygous recessives in nature. So then you get some gene flow between these two populations. So individuals are moving. Some individuals are moving from the first population into the second. They're mating with the individuals in that second population and having offspring. Some of their offspring are heterozygotes. And you also have movement the other way. You have some of these homozygous recessive individuals from the second population moving back into the first population and mating there, and some of their offspring are again heterozygotes. So now that you have gene flow between these two populations and the populations are mixing, the diversity of each population is actually going up. The two populations are becoming more similar to each other genetically, but the diversity of each one is increasing because you started with very low diversity in each one. So gene flow can increase genetic diversity. It could also decrease genetic diversity. It just depends on the specific situation. It will for sure make the two populations more similar to each other genetically. It could also be a positive thing or a not positive thing. So if you have like small isolated populations, gene flow between them is probably going to be a positive thing. It's probably going to increase genetic diversity, and it's going to reduce inbreeding. So it will increase fitness as well. So for example, like with those panthers in Florida, when they brought in panthers from Texas to mate with them and prevent inbreeding, that was an example of gene flow. That's gene flow that we've kind of caused to happen because we're trying to conserve the Florida panthers. You also have a similar thing going on in the lab. If you work with animals in the lab, like maybe mice or rats, or probably even fruit flies, so you're going to have this population of that research animal that you're maintaining in the lab. You're going to be breeding them together to make more offspring that you can do experiments on, and you're going to need to keep track of like who's mating with who and who's related to who. And periodically, when you're maintaining a population of research animals like that in the lab, you have to bring in outside individuals. You're going to have to go and buy some more mice or some more rats that aren't related to the animals you actually already have. That way you can prevent inbreeding and improve genetic diversity and fitness. So that's gene flow, again, that you are orchestrating between populations. But sometimes gene flow can actually be a bad thing for fitness. So for instance, with trout, you have trout that live in the wild. You also have captive trout that are just bred on farms, and they never really make it to the wild or live in a natural state. So if you actually take some of those captive fish and release them into the wild, and they breed with wild populations, that's actually a bad thing for the fitness of those offspring. So this graph is showing kind of the fitness of wild trout when they breed with other wild trout. That's just defined as being 1. That's our basic baseline fitness. You could go up or down from there, potentially. When captive trout breed with other captive trout, but they're raised in the wild, not on a farm, with all of their needs met, they have much lower fitness. They're really just not good at living out there in the wild. When you take a captive trout and breed it with a wild trout, now you're getting fitness that's kind of intermediate between the two. So these offspring are doing better at living in the wild compared to just captive fish, but they're not as good at living in the wild as just the wild fish. So if you were to have gene flow going on between captive populations and wild populations, that would actually harm the wild trout populations. Another thing I'll mention is that through time, technology gets better, transportation improves particularly. Well, for this case, that's particularly important. Basically, we started out with human populations that were pretty fragmented across the world. There's always been migration of people between places, of course, but, it just wasn't that easy to move, especially if you're talking about crossing an ocean or crossing a very large distance in the past. Now it's very easy to do that. So we have a lot of gene flow going on between human populations now that we didn't have in the past. It's one of the main features of like modern human evolution. It doesn't affect every population the same way. Some populations of humans are still quite isolated from the outside world, but we're linked to populations in every continent of the globe now by our technology and by gene flow. So next we'll talk about mutation, which is the only way to get new alleles. It's the only source for new genetic diversity. It will always increase genetic diversity. And there's other things you can do, of course, to increase genetic diversity in a population, but that's all working with alleles that you already have. The only way to get actual new alleles, new genetic information, is by mutation. And there's 2 basic types of mutation. It could either be a point mutation, where a single nucleotide is changing, or a chromosome mutation, or a whole segment of a chromosome is changing. That could involve a whole portion of a gene, or multiple genes, or even an entire chromosome. There's a lot of different options for chromosome mutations. So these are always random changes. Mutation is always random. How it impacts fitness and survival and reproduction, that's also random. Usually it's actually kind of neutral. Most mutations won't have like a significant effect on the fitness of the individual who has that mutation. Some mutations are, of course, really bad to have. Some of them are just a little bit bad to have, and some of them are good. So most common for them to be neutral, then the next most common probably is for them to be bad, and the least common is for them to be good. But, of course, there routinely are good mutations, beneficial mutations that arise, and are then selected for by natural selection and other processes. And of course, whether a mutation is good or bad also does depend on the environment. So for instance, if you have a mutation that makes an animal an albino where the coat doesn't have color all of a sudden, Well, that's a bad thing if they're living in kind of a normal like forest or something. It makes them stick out really strongly to predators. But if we're talking about an animal that lives kind of in a snowy area, that's actually a good thing to be albino, potentially. I mean, it certainly helps you blend in. How common it is for mutations to occur really depends on the species. For large organisms like humans, they're pretty rare. The mutation rate is pretty slow. For humans, it's estimated our mutation rate is about one mutated gamete per 10,000 gametes, or one mutation per 10,000 gametes. So one mutation per 10,000 eggs or per 10,000 sperm. So that's pretty low. Of course, you can get mutations in other parts of your body. Like, for instance, if you started out with not many moles on your skin, and now you look and you spend a lot of time in the sun without sunscreen and you have a lot more moles now, those are mutations that you're looking at. You know, those are all mutations. But they're not going to get passed on because they're in your skin. That's not going anywhere. Only mutations in the eggs, in the sperm, or in the cells that are directly dividing to create eggs and sperm. Only those mutations can be passed on to your offspring and actually affect evolution. So that's for humans, the mutation rate is pretty slow. But if we're looking at other animals, especially, or other organisms, especially small things like bacteria, archaea, or viruses, the mutation rate is much faster. Also, if we look at cancer cells in humans, the mutation rate is much faster than 1 in 10,000 cells. Cancer cells will mutate quite quickly. Some viruses are mutating like so quickly that every new virus actually has mutations. So an example of that would be the flu virus, actually. It mutates so quickly that every single new flu virus has, on average, 2 to 3 mutations in it, compared to the original that it was built off of. So let's see, this graph here is just kind of showing the randomness of mutation. Each one of these lines here is a different population of E. coli bacteria that are raised in the lab over thousands of generations, which a generation for E. coli is like 4 hours long, so really short. And just the number of mutations that you start to see in those populations over time. So you can see that there was one population there that hardly mutated at all, even after 50,000 generations. There was only a few mutations. But most of these populations that they looked at had a lot more mutations than that. And, one of them even had like 2,500 mutations after 50,000 generations. So, even within a given species, the mutation rate can vary widely. And that's partly because it's just literally random chance. Whether you get mutations or not is just random chance. There's, of course, certain things like certain chemicals or UV radiation that will cause mutations, but without those things going on, it's just random. There's also a type of mutation I wanted to mention, which has been important in evolution, which is the gene duplication. That's a type of chromosome mutation where part of the chromosome is copied, and any genes that were in that section of the chromosome, they're going to get copied. So now you have multiple copies of those genes, and they can evolve independently. They can get, mutations independent of each other and take on new functions. So an example of that is this SWS2 gene in fish. It's found in zebrafish and salmon. So this gene encodes a light-sensitive protein. It actually will change shape in response to blue or violet light, and it's something that helps fish see. So you get, through evolution, you actually at some point had a mutation, a chromosome mutation involving this gene, which was a duplication event. So you ended up with 2 copies of the gene. And then they both started mutating independently of each other. So you have your SWS2B and your SWS2A version of those genes. So those are now totally different genes. Each one of these genes would have its own alleles. There's still, both of these are encoding light-sensitive proteins, but the actual wavelengths of light that each one will respond to are going to be different. So there's certain fish that will have both of these copies. More evolution goes on. You actually had another duplication event for this A gene, resulting in 2 genes, A beta and A alpha. So now some fish have 3 of these genes that all came from the first gene originally. They have SWS2A beta, SWS2A alpha, and SWS2B. Each one of these is a light-sensitive protein with a different sensitivity to different wavelengths of light. So this is allowing these fish to see different wavelengths or respond to different wavelengths of light. Then, as evolution continues, you can also have deletion mutations, where a whole section of the chromosome is deleted. And we start to see that some of these genes are being deleted from certain lineages of fish. So, for instance, if you're a sole fish, back in your evolutionary past, your ancestor used to have all 3 copies of these genes, but you lost 2 of them. All you're left with is the A alpha copy. If you're a pufferfish, you're only left with the B copy. If you're a goby, you're left with the A beta copy and with the B copy, but you lost the A alpha copy. So stuff like that can have a significant impact on the evolution of these animals, because you're going to have different functionalities in these different genes and different patterns of who has what gene, depending on who inherited what mutations from who. The last thing we're going to talk about in this chapter is lateral gene transfer, which is also called horizontal gene transfer. So this is transferring genes from one organism to another, but outside of heredity. So not from offspring, or not from parents to offspring, but between individuals who are actually not related to each other and not mating with each other. Horizontal gene transfer is always going to increase genetic diversity in the individual that is receiving this new DNA. So they're always going to get some gene that they didn't have before, or potentially multiple genes that they didn't have before. That could be a good thing for them, could be a bad thing for them, could be a neutral thing for them. Commonly is actually helpful for the individual, or at least neutral for the individual receiving these genes. This is especially common to happen in microbes, which would be little organisms that you can't see with your eye because they're too small. And especially for prokaryotes, so organisms without a nucleus, mostly bacteria. Archaea can do it also. So yeah, horizontal gene transfer, actually, as we're going to see later in the semester, has had a significant impact on the evolution of eukaryotes and even large organisms like plants, but it's most significant for microbes. And its effect on things like plants is mostly because they descended from microbes. Does horizontal gene transfer really count as a mutation? Some people count it kind of as a type of mutation, but I wouldn't really count it as a mutation. It doesn't make new alleles. It gives you new alleles that you didn't have before, but it doesn't make them from scratch. It, is taking them from someone else, someone that you weren't related to, and giving them to you. It could potentially create a new allele if this new DNA that you're getting inserts in the middle of a gene. Then it's going to change the sequence of that gene. That will technically create a new allele. It's not going to be something that's functional, but that could be considered as creating a new allele. But apart from that, if it's inserting between genes, it's not going to make anything new. A lot of the time, actually, horizontal gene transfer will involve plasmids getting passed between bacteria, so little circles of DNA getting passed around between bacteria. Certainly no mutations involved there. I mean, you could have mutations on those plasmids, but that would be separate from the horizontal gene transfer that's going on, which is just moving those plasmids around between individuals. So I said that it's mostly important for prokaryotes and microbes. That is true. It does occasionally happen, though, in larger organisms. So here you have an example of the impact of horizontal gene transfer on the evolution of aphids, actually. Aphids started out not having a color. Usually when we see aphids now, they're green. Well, I should say when I've seen aphids, they're usually green. Sometimes they're gray. Actually, have I seen the red one? I can't even remember. I don't think I've seen the red ones. But these genes are actually giving the pigment to the red aphids and the green aphids. They don't come from aphids. They come from fungi. Those are fungal genes. And they code for enzymes that will create carotenoid pigments. So these 2 bars here, the blue and the purple bar, those are both genes for enzymes that will manufacture carotenoid pigments that will create the red color or the green color. So at some point you had these genes for carotenoid synthesis transferred from this fungus into a gray aphid that had no pigment. After that point, it had suddenly genes to make pigments, and suddenly it would be a colored insect. time goes by, and you can see we have a lot of duplication mutations going on here. So you end up with aphids that have a lot of different copies of these original genes that were from fungi. And these genes will be mutating, changing a little bit compared to each other. So now the pigments that are produced by each one of these are going to be a little bit different. They're going to have different colors. And for the red aphids, they have all of these genes, but it seems that this particular one is the one that's really making that red carotenoid pigment, because you also had a deletion mutation when red aphids gave rise to green aphids. This particular gene was deleted and the red pigment was lost. So instead you have all of these other carotenoid genes left. They still originally came from a fungus, but they're not producing a red pigment. Instead, overall, the pigments they produce are green. For us today, the most important implication of horizontal gene transfer is in the spread of drug resistance or antibiotic resistance. So you have a lot of microbes living in your gut. Especially when you talk about the large intestine, it's literally just full. It's full of bacteria and stuff. Some fungi and stuff, a bunch of viruses also. Mostly the viruses are eating the bacteria though. They're not affecting you. Some archaea in there as well. Mostly it's bacteria and viruses that are affecting those bacteria. So a lot of these guys are good. They're good for you to have. They digest food for you. They make vitamins for you. If a bad guy comes in, if a bad bacteria comes in, it's going to be hard for that pathogen to kind of set up shop in your intestine because there's all these other guys already living there that he would have, the new guy has to compete with them for resources. So it's overall a good thing to have all these dudes in here. But it's not always a good thing. So, anytime you take antibiotics, all these guys are exposed to the antibiotics that you were taking, most likely. I mean, certainly if it's a pill, these guys are all taking the pill right alongside you. So, some of them are going to be dying when you take antibiotics. Some of them are going to have the opportunity to evolve resistance to those antibiotics. So it's not uncommon for at least a few members of your microbiome, your little community of bacteria in your intestine, to be resistant to some antibiotic or other. And because bacteria can do a lot of horizontal gene transfer, it's possible for them to spread that resistance to other individuals. So if we zoom in here, this diagram is actually showing 3 different methods for horizontal gene transfer. So here you have a bacteria. This is the chromosome. These little dots here would be ribosomes. This little circle is a plasmid. In red there on the plasmid, you have a resistance gene. So this bacteria is resistant to an antibiotic because of this little gene that it has on its plasmid. It can do something called conjugation, where it will actually pass this plasmid on to another bacteria that it is not related to. Now this new bacteria has that plasmid, it has the resistance gene, it can build what this is, just that plasmid being used to make mRNA and then make a protein. And so it can build a protein that is going to give it resistance to an antibiotic. You can also have horizontal gene transfer occur in other ways. In this case, you have DNA just kind of lying around in the gut. So sometimes when bacteria die, the cell will break down, their chromosome will get kind of chopped up, and you'll end up with bits of DNA around just free in the environment. Bacteria will actually, some of them will take that stuff up into the cell, and now they have that DNA. So if this happened, if this little chunk of DNA happened to have a resistance gene on it, now this bacteria has that resistance. This can actually insert into the chromosome of the cell. And here, the last way is by viruses. So this is a virus that is injecting genetic material into this bacterial cell. So here's your genetic material or DNA from this virus. It's possible that this could include not just viral DNA, but also DNA from a different host. This DNA is going to go, we see it here inserted into the chromosome. Eventually that's going to activate. It's going to start being used to build viral proteins and new viral particles. And that virus is going to need to cut its DNA back out of the chromosome from the host. When it does that, it might mess up and take a little bit of host chromosome with it, and then you would have a new viral particle with a little bit of host chromosome in it. So that's kind of where this virus would have gotten that from. It would go find a new host, inject its DNA in there, and then that DNA would have a little bit of DNA from the previous host, which could potentially include a resistance gene. So these things can cause a spread of resistance, especially the transfer of plasmids. It's actually pretty common for resistance genes to be found on a plasmid, and a lot of bacteria can transfer the plasmid from one bacterial cell to the next. They don't even have to be the same species. This could be a totally different species of bacteria that is now receiving this resistance gene on this plasmid. So, you have your members of your gut microbiome living here all the time. That's well and good. You have a few bacteria that are kind of just passing through with your diet. They're going to maybe die. Some of them will make it out in your feces. On their way through, it is possible that they could pick up some resistance genes from a resident of your own microbiome sharing a plasmid with them that contains a resistance gene on it. So this could be a guy living in your gut. This could be a guy just passing through and picking up a resistance gene. If this is a guy that's making you sick, now he's resistant to antibiotics. If he lives and he goes out in your feces and, he gets picked up by someone else, now they have a resistant strain of bacteria that's resistant to antibiotics. So this is a major way for antibiotic resistance to spread today. Pretty difficult to prevent that from happening, honestly. But certainly it's very impactful for us. Okay, so that was chapter 23 on mechanisms of evolution.